Did you heard about DNA? Do you know the structure of DNA? Or else, do you know who discovered the DNA? And even, do you know that how the DNA will be replicate and it forms its another copy? Or else, do you know that what codon means? If not, if you don't know the answer of any one of these, then these series of my videos on molecular basis of inheritance is most suitable for you to understand. I'm Dr. Shivanoor. Welcome you for my YouTube classes. Today we are going to start with molecular basis of inheritance. It is a very important high weightage topic. Every year maximum number of questions are used to ask from this topic. That's why it is very important to know. In addition to that, if you want to really understand the present advanced molecular techniques used to treat the different kind of diseases, then to understand the molecular basis of inheritance is a very important one. Then without wasting the time, let us begin to understand at least today in this class about the basis of history of this molecular base, means the about this DNA, how it is started and all. I think all of you know, students, you heard about the Mendel. Mendel was the one who has carried his work in the year of 1865 to 1863. All about 7 years he has worked on pea plant. He carried the many more hybridization experiments on the pea plant, and based on his all results on pea plant, he proposed the different laws of inheritance. What are those laws? Those were proposed by Mendel then. He proposed the one law, law of dominance, and it is the first law. And he proposed the second law, that is law of segregation. So both these laws are based on monohybrid cross on the pea plant. If you are not thorough with what is the monohybrid cross, how he has conducted the experiments, please first try to understand. Then based on the dihybrid cross, Mendel has proposed the one more law, that is law of independent assortment. But all these laws are not famous by his time. No one has accepted the Mendel's results also. The reasons are many more. Why Mendel has not become popular by his time then? Because communication was less. No one has understand use of mathematical formulas in the biological field. And Mendel was not able to propose the proof for factors what he was explaining. But anyhow, whatever he has told, it is all true, that it is came into the light in the year of 1900. All such results he has published in the year of 1865. But one thing is, he has not told the nature of factor. He was saying that it is a discrete unit. But if you ask the Mendel, what is that discrete unit? Today we know it as a gene. But by his time, he was not at all know that it is a gene. But he used to say that factors are there, they were in the pairs and they will get segregated, one of which will be get dominated, and they are independent to each other, so they can independently separate even in the mixture. So that what he was told. But unfortunately, no one has recognized his results during his time. Remember, my dear students, he has expired in the year of 1884. Till his death, no one has recognized the Mendel's work. But fortunately, in the year of 1900, Hugo de Varis, and one more scientist Carl Scoren, and third one more scientist Schemark. All these three scientists, they have told the same thing. They are all of them are independently rediscovers the Mendel work. But let me tell you students, really I appreciate all of these three figures what you are observing on the screen. All these are really shown the humanity, because during that time if they thought let not Mendel to become famous, they can. But these are the scientists. They told that in the year of 1865 itself, Mendel has worked, and even he got the same results that what we are getting today in the year of 1900. Then the Mendel has become a father of genetics, as the genetic is one more branch which is opened in the biology. It is a branch of biology which deals with inheritance and variation. That particular branch has opened up whence the Mendel has become a father of it, and all of these three scientists are made him to become a father. I appreciate and I thank to all these three scientists. They really shown the humanity to the world. Okay, then in the year of 1902, Sutton as well as Boveri, both of these are, they proposed chromosomal theory of inheritance. I hope you remember that. And they argued to propose it. But finally, they told that whatever the factors which were used by the Mendel, they are nothing but the chromosomes, and factor behavior as well as the chromosomal behavior at the time of the cell division is similar that what they have told. But by their all argument, it is concluded that factors of the Mendel's are nothing but the chromosomes only. Remember students now. This is what a very important point to be think now. This scientist has told factors are there. Of course, we know today. We know more than this Sutton and Boveri actually. But during that time, during the 1900, microscopes are not so evolved. They don't have well advanced equipments to study the DNA. That's why they concluded that whatever the Mendel's factors are there, they were nothing but the chromosomes only. Then by the discovery, even it came to know that chromosomes are not made by proteins as well as DNA. Okay, but further discovery we were observing in the year of 1910. One of the scientists, Morgan, along with his students, he has worked on Drosophila melanogaster. Do you know that? Why the Morgan, along with his students, he has started to work on a Drosophila melanogaster? What is the main agenda of their experiments then? Students, their main agenda to start the work on a Drosophila melanogaster is to give proof for the chromosomal theory of inheritance. Whatever Sutton and Boveri is already proposed in the year of 1902, chromosomal theory has proposed. To that chromosomal theory, these scientists are want to give the proof for that. But of course, they have proposed the proof with some extent, but they have discovered the one more very important concept. What is that they have discovered? They have discovered about the linkage. And later on, even you know that students, one of the Morgan's student is none other than the Alfred Stratton. he has mapped the chromosomal theory of, he has mapped the gene on the chromosomes. Even you can able to observe the chromosomes over there on the screen. On every chromosomes, where that gene may be, that what he has discovered. Do you know that on what basis he can able to discover the position of that particular gene on the chromosomes then? It is completely based on the percentage of recombinants. My dear all NEET aspirants, you should know that he did the chromosomal mapping by using percentage of recombinants. But by using the frequency of recombinants, measured the distance between the genes and mapped their position on the chromosomes. Dear students, later on, many more new different techniques are developed, and with the help of all these new different techniques, they come to know that DNA is the genetic material, not the protein. But remember that many scientists were arguing that why can't the protein is a genetic material? They were asking, okay, DNA you are all saying it is a genetic material, but some other scientists were arguing that why can't the protein is the genetic material? Okay, let us now further discuss why the DNA is going to be genetic material, why not the protein? Why we won't consider the protein as a genetic material but the DNA? What property that genetic material should have then? Let us first discuss here what are all the properties that genetic material must have. Either it may be the DNA or it may be the protein or it may be the fat, whatever it may be. But we want to know that what must be the property of that genetic material, because by this time much more advancement is occurred in the scientific. That's why they have already know that what kind of the genetic material must be, what property it should have. Okay, then let us discuss one by one all the property of the genetic material. And later on we'll see that whether these properties are going to be fulfilled by the DNA or whether not, or whether these are going to be fulfilled by the protein or fat. We want to discuss that later on. First of all, here we'll discuss about what property that genetic material should have. Dear students, genetic material, it should found in all the cells of the body. That is one very important thing. It means that whatever the genetic material DNA, let us say the DNA, let us say the protein, it may be, we were not concluded now whether the protein is a genetic material or the DNA is a genetic material. Many scientists were saying that the DNA is a genetic material, but some are have a doubt whether the DNA is a genetic material or the protein is a genetic material, because whatever the chromosomes are there, students, those chromosomes are made by the protein as well as the DNA. That's why they have a confusion now. And it is also not very easy to answer that particular question, students. One side, develop equipments were not so developed. One side, we don't have a much more advanced techniques to discover that. But anyhow, it is a challenge for the scientists to know that whether the DNA is a genetic material or the protein is a genetic material, whether the DNA is fulfilling the characteristics of the genetic material or the protein is fulfilling all that characteristics of the genetic material, that what they want to discover. Okay, but whatever that genetic material must be, it should found in all the cells of the body. You might be thinking, Sir, whatever the RBCs are there in our body, they will not at all have the nucleus itself. If the nucleus is not there, 100% there is a no DNA in that. No chromosomes, nothing, no DNA. They are eunucleated cells. If it is the condition, then how can we are able to say that the DNA is the genetic material? Okay, let it be your doubt. But remember, my dear students, RBCs are even had a nucleus during their early development. Whenever they are just formed in the bone marrow by the process of erythropoiesis. By that time, RBCs too have the DNA genetic material, that is the chromosomes are there, everything is there inside the RBC. It is just like the normal rest of our body cell. But during the maturation, RBC is going to be lost its DNA complete nucleus. It is going to become an eunucleated cell, and it is for the purpose. It wants to provide the more space for accumulation of hemoglobin molecules. Let it be, my dear students. But remember that genetic material, whatever we want to see, that genetic material, according to the many more scientists, it must found in all the cells of the body. Okay, but I told you one exception is that for the DNA is not there in the RBCs. But I told you it is not there, of course, but during after the maturation it is not there. Before the maturation, even the RBC is like the rest of our normal body cell. So then another property, it must found in equal proportion. Okay, one thing, it should found in all the cells of the body. Second one is it must found in a equal proportion. Suppose if one of our body cell have a 46 chromosomes, even all the cells of our body must have 46 chromosomes only. It should not be like one have an 46, another have a 50, one more have an 20. No, it will not be like that. It never going to be like that. If it is the situation, then probably that organism is no more going to survive. Okay, that's why what we were observing here, whatever the genetic material we were expecting, that genetic material must found in a equal proportion in all our body cells. Okay, third property, it should found equal amount in the individuals of the same species. Yes, this is also an important one, students. Only observing the equal proportion of DNA in every cell is not important. Okay, of course it is important, but along with that, even the every organisms of the same species. Suppose, for example, if you are a human, every humans on this entire planet. If it is the crow, every crow on the entire planet Earth. If it is the mango plant, then everywhere throughout the world, whatever the mango plants are there, they must have a same amount of DNA in their every cells. Okay. Then another property, the fourth property, it must keep the genetic information in a coded form. Yes, in this chapter, later on, we were going to understand what that codons means in the DNA. We'll see that even by that time, whether the DNA have a codon or the protein have a codon, that also we were going to discuss later on. But remember here, students, the fourth property we were expecting for the DNA, it must keep the genetic information in the coded form. Okay, by taking a short break, I am requesting here to you all subscribe my channel, like the videos and share with your friends. I used to explain the entire biology in a well prepared animated PPTs in the same way what you are observing right now. I hope it is becoming useful for you all. Please do subscribe. If not yet subscribed, please do it. And if you already subscribed, please like it. And if you already liked, please do share this video along with your friends. Okay, let us come back again to our discussion over here. We have observed here the total 4 properties of every genetic material. The 5th property is it must have more stability. Oh, that is also an important one, students. Suppose if it don't have a stability property, then it may degenerate or it may keep on subjecting to the mutations. That's why we were all expecting that whatever the genetic material we were going to understand, that genetic material must have more and more stability. Right? Later on, we'll see that whether the protein have a more stability or the DNA have a more stability or RNA have a more stability. Which one is really have a more stability? That what we will check it out later on. But first here, we were going to understanding just what the property that every genetic material should have. Okay, the 6th property, it should pass the same information to the next generation. Dear students, if your parents are not passed the same information to you, then how can you looks like the human? If you are looking like the human, if you are a human, if you have a same nose at the same location on your face, if you have a eyes, if you have a eyelids, if you have a fingers, hand, every organ, like every individuals, because whatever the genetic material we were expecting, that genetic material is the one which is passed the information in the exactly same form to you, and 100% you too are going to pass the same information to the next generation without any alteration in that. Okay, 7th property, it should permit the little variation and the recombination while the sexual reproduction, my dear students. Okay, of course it should have a more stability, no doubt in that, but it should permit the very slight variation in that, slight mutations in that. If not the mutations, just think a while. If not the mutations, if not the any variation, then how can you are expecting the evolution process? Evolution is occurring. It is going to occur continuously. It is a continuous process. By the process of evolution only, we are all evolved. By the process of evolution, every organisms on this entire planets are formed. How can it is all? It is because the whatever genetic material we were expecting or it is there, it is permitting the little bit variation in the while passing the information from generation to generation. So last one more very important property, it must have the ability to duplicate itself. Yes, the DNA replication we were going to understand. Probably most of you might be already covered this topic. Have you gone through anywhere that the protein is going to be get duplicate itself? No. But the DNA, that's why we were observing, many scientists were arguing that, or they are accepted that the DNA is a genetic material. But some scientists have a doubt. Okay, that is what our main intention over here, to make clear their every doubt. How the scientific community has tried to clear their doubt, to explain them protein is never going to be a genetic material but the DNA. Okay, what the last property we were observing here? It must have the ability to duplicate itself. Okay, these are the property we were expecting in the genetic material. We want to know that whether all these properties are going to be fulfilled by the DNA. Okay, let us ask this question then. Let us see that. Okay, the first property, it found in all the cells. 100% DNA found in every cell except the matured RBC. Okay, let it be. But even it had a DNA during its development. Okay, that property is going to be fulfilled by the DNA, no doubt. Second property, it can duplicate its genetic material. Or DNA can able to duplicate its entire material. No doubt in that. Means the DNA replication occurring. It can permit the variation or the mutations. Yes, that is also expected for the DNA. No doubt the DNA will be subjected to the mutations, or it may be subjected to the variations during the sexual reproduction. It must be more stable. Oh, DNA is much stable genetic material, students. We'll see even in detail how this is going to be a most stable genetic material. Remember, my dear students, suppose for example, if in a crime cases, one of the person has burned up suppose completely, even in that ash of that burned person, we can able to extract out that DNA. In such a high flammable fire also, the DNA can be stable. But we don't know whether the proteins are stable or not. We are going to understand it later on. Right? And the next property, all the somatic cells of the body contain the same amount of DNA. That is 100% true with respect to DNA. DNA is equal in every cells of our body in a particular species. Suppose for example, the Homo sapiens sapien, if you taken, every species of this Homo sapiens sapiens, means every individuals of this Homo sapiens sapiens are have the same amount of DNA in them. Okay, this is also going to be fulfilled by, this property is going to be fulfilled by our DNA, no doubt. It can be stored the genetic material. Yes, the DNA is going to be stored the genetic material, students. No doubt in that. Okay, all these properties are going to be fulfilled by the DNA. Let us see now whether the protein is going to be fulfilled all these properties or not. All these properties are fulfilled by DNA. But we want to know that whether the proteins are going to fulfill or not. What I am written here, but some other scientists were asking why not the protein or the genetic material. We'll see that now whether the properties of the genetic materials are going to be fulfilled by the DNA or not. If the DNA is a genetic material, then tell us the structure of the DNA. That is also there. And one more question for the scientific community, because many of them are saying that the genetic DNA is going to be a genetic material, but the other group of scientists Some other scientists were asking that why can't the protein is going to be a genetic material? That is their first question. Second question is, if the DNA is a genetic material, then can you please propose the structure of DNA? That is what their question, students. We'll see that how the scientific community has tried to answer both of these questions. First of all, we'll check it out whether the protein is going to be fulfill the properties of the genetic material or not. Okay, the first point, proteins are found in all the cells of the body. Oh, this is going to be fulfilled by the protein even. We were observing that the genetic material is going to be found in all the cells. Of course, the protein is also found in all the cells. DNA is also found in all the cells. Protein is also found in all the cells. But remember, even RBCs too have a protein. Of course, the matured RBCs don't have a DNA, but they have the protein in them. Means even RBC is not also an exception for them. Matured RBC is also not an exception for the protein. All the cells are going to be fulfilled this particular properties of the genetic material. Second property, it must diversed bio macromolecule. Oh, it is also a very diversed biomacromolecule. Protein is a very diverse macro biomolecule. It is there in every cell. Proteins are the macrobiomolecules and found in a different varieties. Remember, my dear students, what we were observing here. Proteins are the one who have a very diverse form, and they even found in every biomolecules. Means these are the properties which were made those other scientists to expect why the DNA is not going to be, protein is not going to be a genetic material. Of course, the protein, they can carry many more specific functions in the body. Remember, my students, what is the reason why they are saying why not the proteins? That what their question. The reason for the question is they will know that the proteins are the macromolecules. Proteins are present in a very diverse form. Proteins can carry the many more specific functions of the body. These are the reasons why they were saying that the protein is the genetic material. But remember, my dear students, proteins do not have the mechanism of their duplication. Another property they are not able to fulfill. They will not able to store the genetic material. These are the two reasons, my dear students, made the different other scientists to understand that protein is never going to be a genetic material. What are the two properties we are saying here? They do not able to duplicate, and another reason is they will not be able to store the genetic information. If they are not able to store the genetic information either in the form of anything, either in the form of coded or it may be any other form, but what is the use of this protein to accept it is a genetic material? That's why many scientists were said genetic material is 100%, it is going to be a DNA only. Okay, here we have concluded finally the DNA is the genetic material. But later on, even many scientists were carried the different kind of experiments, finally proposed with the experimental basis also. Protein is never going to be a genetic material, but the DNA. Even all such a experimental discovery also we were going to understand in the same topic. Okay, let us some other data, statistical data we will get here. It is very important to know as a biology students. Cosell, in the year of 1801, he isolated 5 different nucleotide bases from the cell. See here, it is before Mendel. Mendel was also not know what the genetic material is and how it is. He will not know the factors also. What are those factors? But before him, in the year of 1800 only, Cosell has discovered 5 different nucleotide bases. What are those 5 different types of nucleotide bases then? He discovered one is adenine, thymine, guanine, cytosine and uracil. These are the 5 different nucleotide bases he was already discovered in the year of 1801. Okay. Then the Friedrich Mischer, about his discovery, we were going to need to know that, students. In the year of 1869, he was the first person who has isolated some genetic material, acidic substance first identified in the nucleus of the pus cell, and he even isolated from it. And he called that particular acidic substance as a nuclein. Remember, my dear students, who given the term nuclein, if they ask, is none other than the Friedrich Mischer in the year of 1869. It is mentioned even in the NCERT. You can read it out. And later on, after almost about the 10 years later, Fleming in the year of 1879, he is the one, he told that whatever the nucleic acid was already discovered, nuclein was discovered, that acidic substance in the nucleus is given the basic stain and found it is the thin thread-like compound, and to that he called chromatin. My dear students, you should know that inside the every cell, one which is not going through the division, it have chromatin forms, not the chromosomes form. If you don't know what the chromosome is, chromatin is, then you can check out my one more video, which you probably observing in the form of eye indication above. You can go through that video. You comes to know that how that entire genetic material in the form of where the chromatin is there, where the chromosomes are going to form, that I have in detail explained in that video. You can check it out. So in the description of box also, I will give the link of that video. You can go through that, students. Okay, fine. Let us continue in this video then further. So then in the year of almost about 10 more years later, 1889, Altman was the one who had renamed nuclein or the acidic substance as the nucleic acid. Remember that. So they may ask you who given the term nuclein, who given the term chromatin, who given the basic stain to the nucleic acid. So this kind of the questions they may ask you, students. Except the first one, rest whatever the 3 scientists figures you are observing, faces you are observing, all these faces are there in the NCERT, my dear students. Friedrich Mischer's explanation is there. Even Fleming's explanation is there. Even Altman's explanation is there. Remember all these 3 scientists. Look their beautiful faces, how wonderfully they have discovered the things. And remember that what terms they have proposed already. But it took many years literally to conclude whether the genetic material is the DNA or the protein of the chromosome. Got it, my dear students? Okay, we were studied the properties right now based on all the discoveries. But to even answer such kind of the properties, to validate those properties also, really it took the many years for the scientific community to explain about it. Okay. Then let us begin to understand regarding the nucleic acids. Remember that the nucleic acids are present in the two different form. One, it is present in the form of DNA, of course, and another one is in the form of RNA. D-ribose nucleic acid, ribose nucleic acid. Remember, DNA, it acts as a genetic material that according to the most of the scientists, okay, that is true also even based on our advanced knowledge, we can say that it is true also even. But remember, RNA is also act as a genetic material for some of the organisms. You probably heard about RNA viruses. One of the RNA well-known virus is a HIV. They are the retroviruses even. They have reverse transcriptase, one more enzyme in them that whatever the RNA molecules are there, they are going to be becoming an DNA after the infection. But remember, DNA is not only the genetic material for every organisms. RNA is also an genetic material for at least some of the organisms. Okay, it is DNA is genetic material found in most of the organisms, but RNA is found as a genetic material in some of the viruses. But the RNA can be able to carry different kind of other functions. It can act as an adapter molecule, even it can act as a structural molecule. So these kind of this other additional functions this RNA is used to carry out. Fine, are you clear over here? Later on, we were going to even understand the different experiments. Based on that, how can the scientists are able to say that prove that the genetic material is a DNA only, it never going to be a RNA or even it never going to be a protein. We'll see later on. But remember whatever we were discussed over here till that, and even whatever the RNA is there, third point you can observe on the screen, it act as an catalytic molecule. RNA act as an adapter molecule, structural molecule, catalytic molecule. So these are the additional properties even along with the genetic as a genetic material for the some of the viruses, but the RNA is going to be a adapter, structural molecule or the catalytic molecule even in us also. Okay, then what are all the points we were going to cover under this topic then? What is the plan of NCERT board then? In this chapter, mainly we were going to understand the structure of DNA. You're probably observing one of the very beautiful animated DNA on the beside. About this entire detailed structure we were going to understand in this topic. And we are going to understand the DNA replication. How can this DNA is going to be replicate then forms the one more copy. We are going to understand about the transcription process, means from the RNA from the DNA how RNA is going to be formed. And even in this chapter we were going to understand that genetic code I have told you just before. What is that genetic code, that also we were going to understand. And one more point about translation process also we were going to understand. From the RNA, how can the protein is going to form, that also we were going to cover in this chapter. And one more point, it must be 6. One more point we were going to understand in this chapter is about the basis of regulation. All these details we were going to understand. Under this V1 we are going to cover about different operons also, lac operon, that kind of the operons we were going to cover. And even we are going to understand regarding human genome projects also. Okay, all that things related to the genome. My dear students, again I motivate you, please try to understand the DNA in detail. Otherwise, in your higher education, you literally going to face a much more problem because if you don't know the DNA in higher education, you are not able to perform well. And all my doctors students, I'm motivating you students, in the future you need to know that what this DNA and how it is all made up of. That's why you should very thorough with this. Okay, then where that DNA is actually there, many of the students, those who are all from the rural area, they will not know where that DNA. We will keep on teaching the DNA. DNA is like this, DNA is like this, DNA is having a codon, DNA codes for the many kind of proteins. That what we will keep on explaining. But little it is sad thing to know that no one is going to explain where that DNA is located. Why not? Because it is all covered during your high school only. In a high school classes, they were explained to you about where that DNA is located. Okay, let us see then. Once we'll see that where that DNA is located inside our body, you are observing probably one of the beautiful animated picture of human body. This entire human body made up of the different number of cells. Whatever these all the cells are there, they have a genetic material. Let us zoom out one particular cell over here. Suppose if you zoomed it out, so this is what, and one single cell of our body have a boundary of plasma membrane. Inside it there is a cytoplasm is there. Inside the cytoplasm there is a nucleus is there. And all of you carefully observed on the screen, inside the nucleus there is a presence of a thin thread like genetic material. It is the one chromatin. Suppose if you take out one particular chromatin out of it and observe how it looks like, it is the chromatin. You can see that it is the thin thread like compound. It is the chromatin. After once the flamingo has stained this chromatin genetic material, he called it as a chromatin. Okay, fine. But by this, what you come to know, whatever the DNA we were going to understand, it is there in our every body cells. Whatever n number of cells we have, every cells are have a DNA inside them, except only matured RBCs. Leave about the matured RBCs. Except that all the cells of our body have a DNA in them. Got it very clear? Otherwise, once again, listen, my dear students. DNA is there every cells of our body, not only in us. Even if you take an any plant cell, every cell have a DNA its own. If you take an other animal cell, it have the every cell have the DNA inside it, right? So these where the DNA is present, the DNA is present inside the cells, inside the nucleus of our body cells. Fine. Then let us check out even what is the difference between the prokaryotic DNA and the eukaryotic DNA quickly over here. Whatever this DNA is there, my dear students, this DNA is different in prokaryotes little bit, and it is different in the eukaryotes. Means the packaging process is different only, nothing else. Okay, we'll see here. It is a single circular DNA is there. Many of you might probably know that during the replication, that whatever the single circular DNA is there, it comes in contact with the mesosomes. Okay, then it will duplicate. And here in the eukaryotes, they are found in a many more copies and in a linear form. They are not the circular. Linear means they are all cut pieces are there. And the prokaryotic DNA is not bounded by the nuclear membrane. There is no nucleus. That's why we call it as a prokaryotes, no? And in the eukaryotes, DNA is bounded by the nuclear membrane. In eukaryotes, it is bounded by the nuclear membrane. Okay. Then in again prokaryotes, it is naked without histone proteins. So it is not at all wrapped around the histone in case of prokaryotes. It is just a DNA is there, as a single circular DNA is there. But here in the eukaryotes, it is packed with histone proteins. Remember, my dear students, whatever the DNA is there inside our body, it is packed with the histone protein. That is the reason why many scientists were saying, why not the protein is going to be a genetic material? Because both of them are found in the chromosomes. Then why not the protein? That is what their argument. Okay. But in prokaryotes, there is some other extra circular DNA is going to be found. It is known as an plasmids. But such kind of some other circular DNA is going to be found inside the mitochondria as well as all kind of the plastids. Yeah, this is what the bit difference is there, my dear students. Okay, let us discuss then about the structure of DNA. Okay, my dear students, I am understanding right now. So probably you might be now already it is very lengthened class. So I hope we will stop here our class. In the next class, we are going to discuss about the structure of DNA then. Without missing, properly observe the entire from beginning to end the entire class. That will give you the perfect strength on this particular topic. You are never going to miss a single question from this topic, whatever the number of questions they may ask you. I hope this, whatever the video is there, it brought you the very good knowledge related to the genetic material. If yes, don't miss to press the like button. Please do press it. And if not yet subscribe, you please do subscribe, my dear students. It is never going to be cost you anything, but it really matters more for me. It motivates me to do such kind of videos further, and also even it is motivating me to work for you all peoples. Okay, once again I thank you all, my dear students. You please comment what you are expecting me in the next video, and on what particular point you are expecting me to do the videos. Don't at all hesitate to comment. 100% I will try my level best to give you the best. Fine, let us end our video then, students. Thank you once again. Thank you, my lovely all dear students. Have a great day.